This method can help answer key questions in the chloroplast field. For instance, the composition of the chloroplast protein import machinery. It is essential for greening and photosynthesis and hence for food production on the planet. The molecular machine consists of two separate complexes in the outer and inner membrane of the chloroplast called TOC and TIC, but their composition is not fully known.
The main advantage of this technique is that the purification of Arabidopsis TOC-TIC complex can be accomplished in a single-step purification method, TOC-TIC purification. It is a specific and efficient method.
Generally, researchers new to this method may struggle because they're not familiar with the isolation of chloroplast membrane fractions, detergent solubilization, or affinity purification.
After preparing the Arabidopsis plants, grind 10 grams of 3-week-old seedlings in liquid nitrogen using a cold mortar and pestle with sand. Add 20 milliliters of cold grinding buffer to the ground tissue, mix well, and let the mixture thaw on ice. Next, soak 2 layers of quick filtration material with grinding buffer. Filter the homogenate through the 2 layers of filtration material into a 50 milliliter conical centrifuge tube. Centrifuge the filtrate at 1500 times G and 4 degrees Celsius for 10 minutes and transfer the supernatant to a fresh, pre-chilled